puberty and all the hormones and all these pathways. And what we really wanted to do is talk about the biology. What we really want to do is use that tool. And y'all know Marvin, right? Let's get it on like Marvin Gaye, that song. Where's the real Marvin? And I feel like there's been all this build-up. There's been all this questions that you guys want to ask and things that we wanted to talk about. And this song, if you don't know, Let's Get It On. Exactly how it feels, this part of the course. So as you know now from the syllabus, we're going to start to now talk about life, reproduction. Male reproduction first, female reproduction. We're going to start with the male and the female. We're going to date, get to know each other, copulate, inseminate, ejaculate, then use birth control, so impregnate. We're going to take, it takes 9 months, but we're going to take just a couple lectures to gestate, heart rate, and of course you got to feed him or her, so we're going to lactate, grow him up, graduate, matriculate. All these things involve hormones. So let's get it on. Because I know you. We're going to talk about the anatomy first. This is like a weird thing, right? Like just even saying the word. It ain't even got to be funny. You just say the word and it makes you laugh. Why? Whole different relationship men have. We name them. We talk about them as if they're friends. We draw pictures of them. Tell the truth. How many guys have seen graffiti of a penis drawn on the bathroom wall? Okay, how many women? See, we're drawing them. It's us. We make songs about them. Lil Wayne said he can't make a song about his because the story would be too long. I think the funny thing is, if you like tattoo the penis and put a wig on, it'll look like Lil Wayne. We're going to start with the anatomy. And then we're going to talk about the physiology. We're going to do the anatomy first. Then we're going to talk about fertility and the HPG. And all the hormones are involved. Most of which we've already talked about. Then we're going to talk about erection or arousal and all the hormones that are involved in that. And then finally, ejaculation and all the hormones involved in that. Let's start first. This is a side view. Get all the parts in there. Here we go. This is the glands. That's the shaft. The scrotum. We have one scrotum. Somebody asked me that one year. Marsupials have two. Humans just have one. but it has in it 2 testicles, and a testicle is a testis that's outside of the body. You know, and this is a part of class where I really start going fast. You guys got to slow me down. If I start going too fast, somebody say, stop, slow down, say that again. Okay? This is the inside the testis are the testicular tubules. That's what I've tried to illustrate there. I have some models I'll show you. And inside the testicular tubules is where the sperm are going to develop. Where the testicular tubules come together is a section called the rete testis, R-E-T-E. So there's a whole bunch, I'll show you a model in a minute. There's a whole bunch of tubules that are going to come together to form something called the rete testis. And sort of sitting on top of each testicle is a word that I can't pronounce, the epididymis. Has too many D's and I's. In fact, I don't even know if I can spell it. And the mature sperm that we'll talk about in a second are in the epididymis, not in the testicle. So when we actually ejaculate, nothing's actually coming out of your testicle, despite the slang terms that some of you guys may use for that particular event. I think that's how you spell it.
So what distinguishes that between that and the rete testis?
So the rete testis, I'll show you a model. The rete testis are actually where all the testicular tubules come together. Whereas the epididymis is a structure. Epi means above. It's a structure that's sitting kind of above each testicle. And the actual mature sperm that comes out when you ejaculate is coming from the epididymis, not actually from in the testicle. And then there are 2 glands. Before it's getting crowded. Here, let me write some abbreviations. VD, that does not stand for venereal disease or Valentine's Day. That's the vas deferens. And then there's a paired gland on each vas deferens. SV. It's a gland called a seminal vesicle. And in this gland, it's called the bulbourethral gland. It's also called the Cowper's gland. And you'll see on Tuesday, there's a similar gland in females called Bartholin's gland. They're both involved in secreting fluids and lubricating the reproductive tract. And they're analogous, or synonymous, I guess, glands. Good. Okay, now I'm going to do 2 things. One is we're going to make a section. So imagine I'm like cutting a testicle in this plane. If we do that, So I'm cutting it like this. If you do that inside the testicle, and again, I'll show you a model in a minute, there are testicular tubules. And these testicular tubules are formed by the so-called nurse or Sertoli cells. Americans call them Sertoli cells. I was at an endocrine conference once with an Italian guy. He said, "I hate the way you mispronounce the man's name. The man's name was Sertoli." So I always say Sertoli cells. And hopefully there's hundreds of students now, people all across the country, that pronounce it right. The Sertoli cells, not Sertoli. So these Sertoli cells, or nurse cells, actually form a tight junction to protect the sperm that's developing inside the testicular tubules. So your blood, actually, if you're a guy, your blood has never actually seen your sperm. That's why actually if you get kicked hard enough, you can become infertile, because if you bleed, and you can develop antibodies to your own sperm. Or if you've had a vasectomy, or you have otherwise blood has been exposed to sperm, you can develop antibodies because this got sealed off. And you'll see the same is true for oocytes. This got sealed off way before your immune system developed. So your germ cells have never seen your blood. There's no blood traveling inside the testicular tubules. There's also around each testicular tubule something called myoid cells. Myoid means muscle-like. So these, am I going too fast? So there are these cells, and people think, I don't know if it's true or not, this is what I was taught, people think that these myoid cells kind of contract and move the sperm along in the testicular tubule. I don't know if that's true, but that's what I was taught. So people think these myoid, or muscle-like cells, actually contract and keep the sperm sort of moving along, if you will. Wait, Dina?
Yeah.
Okay.
What do you mean if the Sertoli cells are nurse cells?
Well, they provide nutrients. So the sperm, as you know, has, well, I'm assuming you know, has very reduced cytoplasm. So the Sertoli cells provide nutrients. They, as you'll see in a second, they also provide a hormonal environment that's conducive to sperm maturation and development. But the sperm, the developing sperm themselves are fairly minimal. In fact, they only have like 8 mitochondria or something like that. Then, there's these interstitial cells. Interstitial means between tissues. These are the so-called interstitial cells of Leydig. Since I try to do the Sertoli Italian, I have to try to do the German as well, to be fair. They're the interstitial cells of Leydig, named after a guy named Leydig. We're good? Okay, now we gotta make another cut. Guys, you guys don't like this one. Well, not that you like it. Yo, either. If we make a cut this way, stop it. And so now you're looking at the penis like this, like straight on. There's the urethra. So that's the common hole from which semen and urine comes through. The urethra is surrounded by something called the corpus spongiosum. Corpus means body. Spongy means spongy. So it's like a spongy tissue. And then there are two sets of tissues that run along the dorsal side of the penis. Called the corpus cavernosum, or the cavernous body. And it's these tissues. I'm going to do an illustration. I mean, not an illustration. I just did an illustration, a demonstration. It's these tissues that are responsible for obtaining an erection. Because these tissues, this cavernous tissue, this spongy tissue, fill up with fluid, not actually with blood, but they fill up with plasma. And that's what is responsible for erection in both men and women, as we'll talk about on Tuesday. So I have a couple of models, because you know I make my own stuff. So I made this like 21 years ago. I'll pass it around. You can see now it's got Band-Aids on it. It's been through a lot. So imagine that this is a scrotum, all right? Inside, oops. Sorry. Oh, that came apart. Give me a second. I hate knives. So inside the scrotum, here are my two testicles. There's a sheath called a tunica around each testicle. And this would be my vas deferens and my urethra. Right, we don't have a bladder on here. And if you take this tunica off, this is what the testicular tubes look like, what I've tried to illustrate. Okay? So there's a series of rings. Sorry, we lost one over here. And then these all come together. When they come together, that would be my reed testis. And the picture I've drawn right here, okay, so this would look like this. The picture I've drawn right here is if you cut it this way. So you're looking down these tubes, and that's where the sperm is all developed. Does that all make sense? All right, here, I'll pass it. Be careful. Like I said, you can see there's Band-Aids and stuff on it. It's been through a lot. Harold? Yeah, there you go. Be careful with stuff. I have another model that you guys can come up and see. I won't pass it around because it's a little delicate. A student sent me this one. But I guess she didn't like mine. Whoa, it's now fell apart. But if you want to come take a look after class, this was actually all within 1 week. 3 different students sent me genitals in the mail. Why are your students all sending me genitals in the mail? One question to answer. You got a question?
So then, like, you say that, like, the tubes come around like this, doesn't mean there's like space.
Say again.
Like, if the tube is curved around like this, doesn't mean there's space between them.
Which two?
The
This particular two. They're almost just like the one that I'm passing. It's not space. I guess it's held together by connective tissue. So it's not like an empty shell like the way I have there. There's things that hold it together. But they're literally, if you look at the model, they're literally rings like that. And the sperm are developing, and the more mature sperm are further up. And the mature sperm that you will actually ejaculate are in the epididymis, not actually the testicles. So an epididymis is where the reed testis and all those tubes come together to form one tube, and then that leads into the vas deferens. There's two, by the way, seminal vesicles. There's one on each vas deferens. There's only one vulvourethral gland, and only one prostate gland. And all 3 of those glands, as we'll see in a second, contribute to the fluid that is the semen. We're good? forget anything? So first let's talk about, we're done with anatomy now. Let's talk about how fertility is controlled. And you've already had, right, we've already done this HPG. So you already know there's GnRH and LH and FSH, testosterone. You already know that those are involved. But let's take a step back and figure out how people know that that axis exists. So a long time ago, people did experiments where if you remove the pituitary, the testes will shrink. You'll have no sperm and no testosterone. So it was a pretty good idea that the anterior pituitary was involved in maintaining reproductive function in males. What's the next experiment you do? Or what's the next group you would test? No.
If you're getting too much testosterone.
No. The next thing I do is I put the pituitary back. Right? You want a negative and a positive control. If you put the pituitary back, you can inject it. So you can literally just inject pituitaries into animals. And you can restore reproductive function. So somebody did these experiments. I can't tell you which, but somebody did these experiments. Now what would you want to know? The question is, how is the pituitary controlling the gonads? What do you do next? What would you do? You're taking a lot of stuff out when you take out the pituitary.
Well, you could take the pituitary out and give testosterone.
Well, I would give. Okay. Well, but let's test the pituitary first. So once you fractionate out the proteins, right, you can do things like you can give LH. And if you give LH, makes no difference. What would you do next? I'll tell you in a minute. You don't. What would you do next? Ask me. What would you do next? FSH. That's what I do next. That's a 5-point answer. So I'd remove the pituitary, give FSH. If you remove the pituitary, give FSH. Then the testicles will grow, you'll maintain the size, but you still have no sperm and no testosterone. What would you do next? Do what? That's what I would do next. That sounds like a 5-point answer to me. Take out the pituitary, give FSH and LH. It seems like it requires both. What's up, Ian?
Can you, like, in these experiments, do they change times? Like, what if it's that you need FSH first and then, do you know what I mean? Like, you need
That's
one
excellent.
and the next one?
That's not correct, but that's an excellent answer. Yeah, you can't know the correct answer. Because the correct answer is, it turns out that even though this is the axis that I gave you, other things that, well, actually, what did the data tell you so far? Your answer was excellent. What did the data tell you? Yeah. Wait, you're sharing. Okay. And you know what, I'm being honest, I can't even see you, I just know you from the seat that you sit in. So, you guys stay in the same seats and I got you. What's up, Sharon?
Well, since the testes grew with the FSH, we know that has some sort of function.
Yeah, so we know that FSH is trophic. I gave you that that's the axis. We know if you put the whole pituitary back in, you get all functions. So all together, what does that tell you? Something else from the pituitary. And it turns out, the reason I said you can't figure it out, it turns out prolactin is required to maintain the LH receptor. Okay? So even though this is the axis, even though you don't read prolactin on the axis, prolactin is required for maintenance of the LH receptor. So if you don't have prolactin in one of your experiments, you don't see testosterone because LH can't bind. If LH can't bind, and I'm going to tell you why. If LH can't bind, you don't have testosterone, which means you don't have sperm. So even though the HPG is that, prolactin is required in order for that to function. Yeah, has a permissive role. What's your name?
Nausea.
Nausea.
But how much prolactin dopamines are always like keeping at a low level?
It is, well, yeah, so prolactin's at a low level, but it's not zero. Without prolactin, you would not function either as a male or as a female. Prolactin is required for LH receptor maintenance in both males and females. So you need an intact pituitary to maintain that. But yes, in general, it's negative. It's going to have a much bigger role after we get pregnant, prolactin, that is. Yeah, your name is? Wonder. I presume growth hormone will do the same thing as prolactin. So the question is, would growth hormone maintain the LH receptor? And I presume, since they bind each other's receptor, they would. Yeah, that's I don't know that, but that would be my presumption. It turns out that testosterone is not actually, well, here, let me show you some data. So it turns out, if you took sperm, immature sperm from the testis, put it in a petri dish, and bathe it in testosterone, those sperm would not mature. It turns out if you did a radioreceptor, see, I can talk about this now, you guys know what I'm talking about. If you did a radioreceptor assay for the androgen receptor in sperm, you get something that looks like this. Which you can convert to a scatron. Hopefully you did. So it turned out until a few years ago, I taught the following. Because if you look at the Sertoli cells, the Sertoli cells have androgen receptor. So people used to think that the androgen was there because it did something to the Sertoli cells, which then maintained the sperm. Turns out I just learned today, what the androgen does is the androgen receptor in the Sertoli cells somehow sets off a signal And the Sertoli cells then determine how many Leydig cells they are. So the Sertoli cells kind of regulate the androgen, which is coming from the interstitial cells of Leydig. What's your name? Demi?
Yeah. Can you repeat that?
Of course I can. The sperm require testosterone. In fact, there's a protein called androgen binding protein, ABP, that's produced by the Sertoli cells. So this androgen binding protein is produced by the Sertoli cells to concentrate androgen in the testicular tubules and in the epididymis. I'll get to what you want me to repeat in a second, Dave. So the Sertoli cells make this androgen binding protein, concentrate androgen in the testis. Androgen in the testis is required for sperm development, but the sperm has no androgen receptor. The Sertoli cells have androgen receptor. And the Sertoli cells respond to how much androgen it is. Actually, you might get the paper this week. So the Sertoli cells respond to the androgen and regulate the Leydig cells and their ability to make androgen. Does that make sense? Still doesn't explain how the sperm finally mature. It turns out, a few years ago, they made you guys know what knockouts are? They made an ER knockout mouse. An ER knockout mice, so they don't have any estrogen receptor. Males are infertile. So it turns out the sperm have estrogen receptor. So the androgen's being concentrated there so that the Sertoli cells can regulate the Leydig cells, Leydig. But it's also being concentrated there because it's a substrate for estrogen production in the sperm. And men who have low aromatase, or mice that have no ER, are infertile. So it turns out the sperm convert that testosterone into estrogen, and that estrogen is required for final maturation of the sperm, and necessary for something called the acrosome reaction. That's what a sperm meets the egg. What's your name?
Minjung?
Minjung.
Yeah, so sperm is responsible for conversion of testosterone.
Sperm has aromatase and the estrogen receptor, and then the final stages of development of the sperm, including its activation in the acrosome reaction, require that they have that androgen, which they convert into estrogen. That was only discovered, I don't know, I want to say 5 years ago, but fairly recently. We're good? There's two other things I want to put on your axis before we talk about erection. Discovered by one of my colleagues here, there is a gonadotropin inhibiting hormone. I don't know how important it is in humans, but it has a role in directly blocking GnRH and LH. And there's also one other compound that comes from the Sertoli cells that's going to be incredibly important in women. Maybe not so important in men, but this compound specifically feeds back on and inhibits FSH. I told you it was going to get a little more complicated. So this compound from the Sertoli cells specifically feeds back on FSH. And men, I'm not sure how important it is. And women, it's the reason why you usually only have one kid at a time, as you'll see next week. Well, we're going to have twins, but I'll explain to you why you usually only have one. Yes.
If the sperm have the aromatase in them, why, when you put the sperm on a petri dish, can't they just convert the testosterone you're getting them into estrogen?
You know, that's a good question. I presume there are other things that are coming from the circular cells. So it's only the final stages of maturation where that's involved. So the androgen has a role in the circular cells when it's in the testicular tubules, but it's the final stages of maturation when they're actually epididymis that you get that. Other stuff? 'Cause let me think for a second. I think that's all I want you to know about that. Other questions? I'll show you a video. So now that's the anatomy. That's how you get the sperm in the first place. Now we need to figure out how to get it out. How many people know how active? Oh, yeah.
Do the sperm cells or the sperm cells themselves have the aromatase or does
Are the sperm cells?
Sorry.
Wait, sorry, give me a second.
Do the sperm cells have aromatase or do the circular cells put it in?
Actually, the circular cells also express aromatase, especially early in development. But in this case, it's actually developing sperm themselves. And I'm actually talking about the ejaculated sperm that are, and I think that's the difference with the question you asked. So I'm actually talking about the mature ejaculated sperm are making estrogen and using that estrogen. And in fact, the acrosome reaction, which is when the sperm interacts with the oocyte, the aromatase and estrogen receptor are required for that to function properly.
So the Golgi sperm don't have aromatase then, only the true one?
I don't know when it appears. No. Sorry, I don't know when that gene gets turned on. Yes.
So is that something that would work in an autocrine fashion?
That is definitely an autocrine. Yes. So they're actually taking up the testosterone, converting it into estrogen, and it's being active inside that same cell. Correct. That's absolutely what that is. I'm going to show you. Who knows how actin and myosin work? Right? You know, maybe I'll just put the video online. We'll take too much time for you to look. But actin and myosin, actin interacts with myosin. Myosin is the one that's a little motor protein. Actin is the thin filament. And the way your muscles contract is that the myosin head interacts with the actin, and that's how your muscles contract. You guys know what I'm talking about? If you don't, I'll post where you can find the video or something like that. That's important because this corpus cavernosa and actually all of your blood vessels and your digestive tract is arranged in the following way. Your blood vessels, your digestive tract, your exocrine glands, all have this myoepithelial layer around an endothelial layer. The endothelial layer is like the soft tissue. The myoepithelium is the muscle layer that can contract or relax. That's how you control your blood pressure. The myoepithelium can contract or relax. And that contraction and relaxation is what controls whether or not you can get it. I'm about to take all the fun out of getting an erection for everybody. Because there are a whole lot of second messages involved that you don't want to think about. It's about to end right now. Let me ask you a question now. If you have an erection And then you'll see the same thing goes for females, for clitoral erection. Do you think your myoepithelium is contracted or relaxed? How many people say contracted? How many people say relaxed? Oh, you guys just saying that because it doesn't seem like the right answer. Because I know, people get this confused every year. So, I have a demonstration. I need some volunteers. Come on, don't be shy. I need a couple of volunteers. Sabrina and Ulysses. You have to come up. You have to be my myoepithelium. So what you guys are going to do is put your hands around this corpus cavernosa. Oh wait, maybe I only need one person. Because your hands are bigger than mine. So if you put your hands around like this, so your hands are the myoepithelium, right? So if you hold tight, if you're contracted, and I try to pump, nothing can go in, right? If you relax, right? Something happened. Okay, you can relax more. So the idea is, the myoepithelium has to relax, and that allows blood to flow into the penis, into the corpus cavernosa, into the corpus spongiosum. So actually getting an erection involves relaxing, not, because everybody always thinks you have an erection, you're rigid, it must be contracting, the muscle must be contracted. It's just the opposite. It's the relaxation that allows blood to flow into the penis, or as you'll see next week, the clitoris. And it's plasma, blood plasma, and I'll show you pictures in a second. It's blood plasma that's actually leaking into that spongy tissue or to that cavernosa and filling it up with blood. Or with plasma, the cells aren't actually going in. Does that make sense? I'm going to tell you how that happens now. Like I said, yeah. Alright, I will. In fact, let me show you this way. First off, arousal starts in the brain. And don't get this confused. Don't get this confused. Oxytocin acts as a neurotransmitter in the brain to stimulate arousal. That's not oxytocin acting as a hormone. It's not leaving your brain and going to your penis. Oxytocin as a neurotransmitter in the brain stimulates a signal that then travels down neurons to stimulate an erection. That's why people who have spinal injury are not capable, injuries in certain parts of the spine, are not capable of getting an erection. Because you have to have a neural signal from the brain down the spinal cord that then stimulates what I'm about to show you now. That's not oxytocin as a hormone. You'll see why I'm adamantly pointing that out next week. That's oxytocin as a neurotransmitter, starts the signal in your brain, which leads to a neural signal that eventually activates a peripheral nerve. Here's what's normally happening in the myoepithelium right now. If you're flaccid, here's what's happening. Right now, myosin is interacting with actin to keep the corpus cavernosa and spongiosum contracted so that blood doesn't flow in. Like you saw when Ulysses was holding the balloon, the air couldn't get in there. Right now, well, I'm making some assumptions, but if you're flaccid right now, this is what's happening. And this is happening because something called myosin light chain kinase activates the myosin. Myosin light chain kinase phosphorylates what's called the light chain of the myosin molecule, and that's what causes the head of this molecule to interact with actin and cause that contraction. You guys have had this in other classes, right? If not, we'll post a video you can look at. I just didn't want to spend the class. So this myosin light chain kinase, let me write your abbreviations, so you'll end up like Dr. Warren. And this myosin light chain kinase is activated by calmodulin. CAM is my calmodulin. And the calmodulin, as you may know from other classes, requires 4 calciums, each molecule. And the calciums come from outside the cell through voltage-gated channels